So the purpose of this video is to be a supplement to the Macromolecules 1.0 video that you watched earlier in the school year. And the idea behind this is that this is going to take more of a molecular approach than what was talked about in Macromolecules 1.0. So if you're looking for, say, the monomer and the functions for most of the major macromolecule groups, you want the 1.0 video. In this video, we're going to talk more about the molecular structure of the four major macromolecule groups and some clues to look for that'll help you recognize those. So it's important that you know the molecular structure for these things, and most importantly, it's important that you know things that are unique about them to allow you to recognize them. So if you were to see the molecular structure for one of the macromolecules and then you had to match it with the appropriate macromolecule group, this video will help you go through and look for important factors that are unique to those individual groups, and it'll help you when it comes to matching them with the appropriate subset of macromolecule. So we'll start off with proteins. We'll go through each group, and the main idea is to talk about, for starters, the elements that compose them. Because when you're looking at the different molecular structures for these macromolecules, elements are going to be something that are a little bit easier to pick out. So for starters, proteins have carbon, hydrogen, and oxygen. This is going to be a big theme because they're found in all of the macromolecules. So those three, even though they can be helpful in some instances, since they're found in all the macromolecule groups, they're not something that are necessarily going to help you narrow down which type of macromolecule it is. Now, the thing that's unique with proteins is they're the only ones that also have nitrogen. Nitrogen technically is also found in nucleic acids, which we'll talk about more in a minute, but there are other clues in nucleic acids that will tell you that you're looking at those. So if you only see nitrogen in addition to carbon, hydrogen, and oxygen, you know for sure it's a protein. Another thing that'll help you is if we're talking about their general structure, it's set up in a linear fashion. So oftentimes when you see proteins depicted, you see them shown in a straight line. There are ways of looking at like three-dimensional models for proteins and how they're folded, but oftentimes you won't come across something like that, at least not at this level of biology. Something else you could look for that are potentially going to be shown in a diagram that you would see of different macromolecules are peptide bonds. Peptide bonds hold the individual amino acids together, so this is something that you could again look for as a clue if you're trying to identify groups of proteins. So if we look at this image, there are two things to look for here. So for starters, we have an amino acid that's depicted over here on the left, and then a dipeptide over on the right. As I mentioned to you before, you're looking for nitrogen as something that's unique. The other thing that will be helpful is in amino acids, they have what's called an R group. Some people mistake this for radon. That R is sort of like the way X functions in math class. If you think of that R as just being a variable group, so there's over 20 different examples of amino acids. That variable side chain is the part of those amino acids that are different. So the rest of this is relatively fixed, but that top part is going to be the part that's changing in proteins. So best thing to look for is you have nitrogen as your one addition to our standard three, to carbon, hydrogen, and oxygen, which you'll see in everything. And then also you have that linear structure in proteins. It's all set up in a straight line. There are no chemical rings in this one, as we see in some of the others. Nucleic acids are the next one to talk about. The elements here are a bit more complicated. So carbon, hydrogen, and oxygen, again, just like we said, are found in all the macromolecules. The things that are unique here is it has both nitrogen and phosphorus. So I mentioned to you before that you would see nitrogen in other things, but you only see phosphorus in the nucleic acids. So that's a good one to use as your clue that you're seeing these things. Another thing that you could use for your advantage when it comes to the general structure is nucleic acids do contain rings. They're actually the only macromolecule group that contains another macromolecule group. So the nucleic acids actually contain a sugar in the middle, which we'll look at the three parts of a nucleic acid coming up in a second with the diagram, but the ring structure is something that you could look for as a clue here. I mentioned before that it contains another macromolecule group. They contain sugar. They also have a nitrogen-containing base, which later on in the year we'll call these nitrogenous bases when we get to genetics. And then they also have a phosphate group, which we sort of alluded to up here at the top when we were talking about them also containing phosphorus. So if we take a second to look at this, they'll always be set up with the same three things. Got the phosphate group on one side, the sugar is always in the middle, and that nitrogen-containing base will be on the other side. So we have nitrogen represented here, the phosphorus then represented in the middle. So those are all good clues. This is the only thing that has phosphorus. So phosphorus is a total giveaway, the only macromolecule, I should say, that has phosphorus. So total giveaway for you if you see that one in there. As far as lipids go, here we're seeing carbon, hydrogen, and oxygen as found in all the other macromolecules. The thing that's unique about lipids is they have two parts to them. Right, so they have glycerol and fatty acids, where a lot of the other ones we're only seeing one part to the different monomers. Now, something to discuss with lipids, they only have these three things. It's only carbon, hydrogen, and oxygen. So you'd think that would be helpful, and it is for the most part, except carbohydrates are the same way. But if you're looking at something and you notice, hey, that only has these three, that narrows it down for you to lipids and carbohydrates. So that'll be a helpful distinction there if you're only seeing carbon, hydrogen, and oxygen, because lipids do not have any additional elements found in them, unlike proteins and nucleic acids do. As far as our general structure goes, it's a linear structure, so you won't see any of the carbon rings that we end up seeing in nucleic acids or in a moment, as you'll see in carbohydrates. They have a triglyceride, which we'll talk about this in the picture in a second here, but it's a combination of glycerol and fatty acids, and that's made through what's called a dehydration reaction or dehydration synthesis. Glycerol, the O in that will give you a clue. If we're looking at the structure here, the glycerol group on the left-hand side is the only one that has oxygen in it. So the part that's shown over here in orange, that is the glycerol. The long chain with the carbons and the hydrogens, that's the fatty acid. Something else to mention is there's two types of fatty acids. There are saturated fatty acids. In those, all of the carbons have a full set of single bonds. So if we look at this carefully, you can see that every carbon can make 1, 2, 3, 4 bonds. In a saturated fatty acid, all of the carbons have 4 single bonds. Whereas if we look down here in the unsaturated one, these two carbons are sharing a double bond. So that's part of what makes this unsaturated. It's unsaturated in hydrogens. You could technically break that double bond, attach another hydrogen here and another hydrogen there, and now you'd have a saturated fatty acid. So unsaturated ones are unsaturated with hydrogens. They could hold more hydrogen than they currently are in their current setup. The last thing to talk about with these has to do with this whole idea of dehydration synthesis or a dehydration reaction. So when the glycerol groups and the fatty acids bond together, there are extra hydrogens and oxygens. So if you look at this one, there's a hydrogen and oxygens. It's called an OH group on the end of a glycerol. We also have one here, right? A hydrogen and an oxygen. So those will combine together to make water as a byproduct. So now if you notice, we have these two oxygens that end up here. But then one of these oxygens ends up in the water molecule, and then 2 hydrogens. So that hydrogen and that hydrogen end up in there as well. So of each of these, I guess you could think of it as being like 3 oxygens and 2 hydrogens. 2 of those oxygens stay, one of them goes, and then both of the hydrogens go in order to make water. So there's extra hydrogen and oxygen when glycerol binds with the fatty acid in order to make a triglyceride. That extra hydrogen and oxygen are just given off as water molecules. So that's what we mean by a dehydration reaction or dehydration synthesis, that water is produced. Last one to talk about are carbohydrates. So the elements here, as I mentioned to you before, are just carbon, hydrogen, and oxygen. So you don't find anything else in there. Something that's unique about carbohydrates is their name ends up telling you the 3 elements that are in them. So the carbo part tells you carbon, and then hydrate is in reference to water, which tells you the hydrogen and the oxygen. So in some ways, carbohydrates are the easiest one because the name kind of tells it to you. The other thing that's really helpful and kind of obvious about these is they contain carbon rings. So if you just have carbon, hydrogen, and oxygen, and a ring structure instead of that linear structure that we were seeing in lipids, you know that you're dealing with carbohydrates. The other thing to talk about is that multiple carbon rings are bonded together with oxygen. And you'll see this up top in our picture. If we look at a monosaccharide, like glucose, so prefix mono tells us one, there's only one carbon ring here. But this one again is all carbon, hydrogen, and oxygen, so we know it's carbohydrate. But then if we're looking over to the right, we have a disaccharide. So this one happens to be sucrose. So we end up in our disaccharide with a monosaccharide on the left, a monosaccharide on the right, and then they're bonded together with oxygen. So that's what this one's referring to down here at the bottom, is that oxygen is the thing that's bonded to hold the different groups of monosaccharides together when we're making disaccharides, or for example, in a very long chain of polypeptides, polypeptides, that would be proteins, of polysaccharides. All of those bonds in between the different carbon rings, those are all oxygens that are holding those things together. So big thing to look for here is that you don't have any extra elements, just carbon, hydrogen, and oxygen. If you have that ring structure and it's not linear like the lipids, then you know you're looking at carbohydrates. So hopefully these tricks will help you when it comes to going through and identifying some of the different macromolecule groups just based on their molecular structure. I appreciate you taking the time to watch, and we'll talk about this more in class.